Let us now discuss photosynthesis. So photosynthesis is defined as the anabolic reaction that uses light energy to drive the synthesis of glucose from carbon dioxide. So this is the source of glucose and oxygen used in aerobic respiration. So this one occurs in the chloroplast, in the eukaryotes, and in the thylakoids of the prokaryotes. So this is the photosynthetic pigment, famously known as the chlorophyll. So it's actually the green color. We usually depict it as the green color in the plant. That's why the leaves of the plants are green. So this, when we say green, by the way, when we talk about the lights, what we see is the light that was bounced off. So that means although it's color green, in reality it's just absorbing other pigments, especially the complement color for green red. It tends to absorb the red color the most, red and oranges color, and reflects or bounces off the green color. That's what we see. So anyway, this is an organic molecule. So you will see here the chromophore. We call it chromophore, chromo, which is the color. This part is what gives chlorophyll the green color. So you will see here a ring, or rather extended rings, and then you have a conjugated system. So review of organic chemistry, conjugated system, you have single double bonds that are alternating. So this is why the chlorophyll has the green color, because of the conjugated double bonds. So that means your electrons, the pi electrons, are moving around, and it's basically the reason why you have the green color. Now, take note, you also have a magnesium here at the middle of your photosynthetic pigment. So that magnesium is actually important for the functioning of the pigment to capture the photons. Now, aside from the chromophore, you have the hydrophobic tail. This hydrophobic tail is the one that anchors the pigment to the membrane. For the case of your prokaryotes, it's the thylakoid membrane, and for your eukaryotes, this is in the thylakoid part of your chloroplast. Sorry. Okay. Now, these chlorophyll, when taken together, so these pigments, they cluster together to form what you call a photosystem. This is used to maximize the absorption of light. So this is the drawing, visual representation of your photosystem. So you have the thylakoid membrane. So, and then this is your photosystem. So they cluster together. So take note, this is a membrane. So you have a double membrane, or rather your bilayer, lipid bilayer. And that is why you have the hydrophobic tail, because you have a transmembrane segment here. So this is the cluster. So you have this is actually photosystem 2. A light harvesting complex. You have core antenna pigments and surrounded by light harvesting complexes. Now, aside from chlorophyll, you also have other accessory pigments. So these accessory pigments enhance the photosynthetic efficiency by absorbing additional photons of different frequencies. So the accessory photopigments here are, well, of course, aside from the chlorophylls, by the way, we have different forms of chlorophyll, A, B, C, and D. So each have different colors. They not exactly give off, they reflect these colors. So you have the bluish green, olive green, bluish green, yellowish green. So you will see here these colors actually gives that's why some plants have darker green, some leaves of the plants have a little bit of other greenish hues. So those are the different pigments, the combination of those pigments. So aside from that, you have the carotenoids. These are the colors of oranges, yellow, brown, and yellow. So usually see them if you are in the higher latitudes when during the autumns, because the light has a different this is one of the more reason is um, maximizing what light is available. So they tend to produce more of these carotenoid pigments to maximize the receipt of the available lights. But if you're in the tropics, mostly the green color overpowers the carotenoid colors. So, but aside from that, from the algae, you have the green algae, the brown algae. That is because the pigment that is abundant in there are different. So it's not a chlorophyll; it's rather carotene or fucoxanthin for the case of the brown algae. So aside from that, you also have phycobilins. So this is the bluish color. These are usually found in the red algae and the cyanobacteria. So these colors, these pigments, takes part in what you call the light reaction. The light reaction is quite similar in a way. You can look at it similar to the electron transport chain in your aerobic respiration. But instead of having oxygen as the final electron acceptor, here you have water as the electron donor. So it's similar, but it's in a bit of a reverse direction. So instead of producing water, taking in or using oxygen as the final electron acceptor and taking and producing water at the end, this light reaction actually splits water. The water is the electron donor, and the final product is the oxygen. So here is your reaction for this one, the water and the oxygen. So this is the reaction for photosynthesis. If you notice, it is the reverse of the aerobic respiration. So you have the products of aerobic respiration, carbon dioxide and water, is actually the reactants of your photosynthesis. And the product of photosynthesis is the reactants of your aerobic respiration. So you might say that this is the reverse of photosynthesis, might be the reverse of your aerobic respiration. But no, but this is the oversimplified view of things. The total reaction might be the reverse of the other, but when you look at each particular step, they are very, very different. So anyway, let's look at this step. So this is the step where water is split. So this part is the light reaction. We usually start the photosynthesis with the light reaction because photosynthesis by nature is an anabolic pathway, unlike aerobic respiration, which is catabolic by nature. So just a review, catabolic breaks down large molecules into smaller molecules and release energy, whereas anabolic pathways uses energy to build up large molecules from smaller ones. And the light reaction part of photosynthesis is the source of that energy they will be using, or rather the plants or the photosynthetic organisms will be using to build up glucose or the food, actually the source of energy to build up the large molecule that they will be building, which is glucose in this case of photosynthesis. Now anyway, how do they harvest energy? How do they generate the energy? So that is why the reason why it's called light reaction is because the source of the energy is the photons from sunlight. So it started with photons, the red and the blue lights, they are absorbed by photosystem. So basically it started with photosystem 2. But why not 1? Photosystem 2 is the one that was discovered a bit later. Photosystem 1 was discovered earlier. So it was called photosystem 1. And then photosystem 2 was discovered a bit later. And so that's why the numbering was a bit reversed. So anyway, so you have the photons of red and blue light. They are absorbed by photosystem 2 and the energy is transferred to a protein called P680. So you have a redox reaction here. And just like the electron transport chain, once that you have the reduction, so when the photon energizes your P680, it splits water. So hydrogen, it gets electrons from water, and you will end up with 2 protons and an oxygen radical. So 1/2 O2, this is the oxygen radical. When you say oxygen radical, you have your oxygen with a non-paired electron. So you have a single unpaired electron here. So it's very, very very reactive. Now, if there's another water molecule that was split on the second instance, the 2 oxygen radicals will be combined to form your final oxygen molecules. So it becomes the 2 oxygen radicals will become O2 if you have 2 waters in the encountering, or rather you split 2 water molecules. So anyway, the electrons that was harvested from the water molecule will be passed around these photosystem complexes. So I won't be I won't be looking into much details, but the end result here is again the formation of a proton gradient. So just like an electron transport system, you have a proton gradient that will be generated. So here the proton gradient is in the thylakoid rumen. So when they pass around the electrons, aside from the proton generated from splitting the water, you have protons outside that will enter as they pass, as the electrons get passed around. And because of this electron gradient, you have an ATPase. Just like in the electron transport chain, you have your ATPase, or sometimes called ATP synthase, that is used to generate the large amount of ATP from the proton gradient. So again, the ATP synthase, how do you generate ATP from the proton gradient? So because of the large amount of protons here, you will generate a basically the gradient where you have a chemoosmotic pressure. We call it chemoosmotic. It's like osmotic pressure, but instead of water, it's basically hydrogen ions. So the hydrogen ions would want, so the protons basically, would want to go to the other side because there are too many of them here. They want to go somewhere where there is not many of them. So the drive of the proton ions to go from the area of high to low concentration, they will pass through this ATP synthase. And whenever they pass through, it will rotate. Once it rotates, it generates ATP for every rotation. So basically, that's your ATP synthase. So end result here is you produce oxygen and also ATP, as well as also electron carriers. So here you will be reducing NADPH as well. And then the second part is the carbon fixation. So this is sometimes called the dark reaction. So in carbon fixation, you get carbon dioxide with the help of the rubisco, RUBPase, or sometimes called rubisco. They will the carbon dioxide molecule will join with a molecule called the ribulose 1,5-bisphosphate. This is your 5-carbon sugar, or rather, not exactly a sugar. You have a 5-carbon molecule that will be joined with the carbon dioxide, so it will become 6-carbon. And this is a very unstable one. It will be split evenly into your intermediate. You have then now two molecules of the 3-carbon 3-phosphoglycerate, and this will be used to build up your sugar. Now this Calvin cycle looks a bit complicated, to be honest. So you have your it's called a cycle because you regenerate the RUBP, the 5-carbon sugar RUBP, ribulose 1,5-bisphosphate. But the way to regenerate it is that you need to have at least 6 Calvin cycles occurring simultaneously. So 6 cycles occurring simultaneously, they are fixing carbon dioxide 1 cycle each. And the end of this one, you will get your pigals. This is your phosphoglycerate. So it's a bit, for example, you have 6 workers. The 6 workers will get carbon dioxide, join it with the starting material, RUBP, and ending up with pigal. So at the end of, for each worker, they will each produce 2 pigals at the end. The pigals is the 3-carbon molecule. So for example, if you have 6 workers working simultaneously, you will end up with how many pigals? So if each of them have 2 pigals each, you will end up with 12 phosphoglycerates. So among those 12, 2 of them will be used to create glucose. And the remaining 10 will be used to regenerate the RUBP that will be used as the starting material for the workers to create new pairs of the pigals. So basically, that's your Calvin-Benson cycle. So it sounds little bit complicated, but generally it should not be. So it's a self-sustaining cycle. So sometimes they visualize it as such. So you have your carbon dioxide. You fix the carbon dioxide using RUBP. And then you will generate PGA. So 6 cycles must occur simultaneously. 6 of them, 6 reactions must occur simultaneously to produce 1 glucose. So you have the 6 carbon dioxide, you will get 12 PGAs. From the 12 PGAs, your it will undergo 1 glucose. So 12 PGAs, you will the of course you will spend ATPs to build up your pigals, and then the ending is 2 of the pigals will form 1 glucose. The remaining 10, because you have 12 pigals at the end of your cycle, the remaining 10 will be used to recycle series of reactions to create the 5 carbon ribulose 5 phosphate. So that is your Calvin cycle. So for the bacteria, aside from some bacteria do not do photosynthesis. So instead they use chemosynthesis. They produce organic or inorganic compounds, or organic compounds or inorganic minerals. They oxidize it to extract energy for their anabolism. Chemoautotrophs, on the other hand, they use carbon dioxide as the primary carbon source, but instead of using light as the source of energy, they use the reduced inorganic compounds as their energy source. And then we have chemoheterotrophs. They use organic compounds, the carbon supplies, and as well as the energy source. So that's it for this lecture on mostly photosynthesis.